Okay, good evening, everyone, and welcome to this special lecture by Professor Herbert Levine. I think it was originally the forever war against cancer, but I guess
it's a slightly modified topic. Same subject.
The same subject, but this is, of course, a topic that, in some ways, all of us, I think, are impinges on all of us directly or indirectly in some way, and so I'm happy that to see all of you here. And I'd like to thank the organizers for suggesting this topic for this lecture, and to Professor Levine for agreeing to give this. I'll now hand it over to Professor Mohit Jolly from IISc, who will give an introduction to the speaker.
Welcome, everyone. It's my great pleasure to introduce the speaker today, who also happens to be my PhD advisor and postdoc mentor, Professor Herbert Levine. He's a university distinguished professor in physics and in bioengineering at Northeastern University, has had a very long, illustrious career in different areas of biological physics, thinking about bacterial colonies, eukaryotic cell migration, and has been working on cancer metastasis for the past decade or so, also thinking about implications in terms of therapy. Herbie has received his bachelor's degree in physics from MIT, and then his master's and PhD also in physics from Princeton. He has been on the editorial board of PNAS, editor-in-chief of Physical Biology, and associate editor of Biophysical Journal. He's also an elected member of the National Academy of Sciences and has mentored a large number of graduate students and postdoctoral fellows who are currently in different roles, both in academia as well as in industry. Without any further ado, I would like to welcome Herbie for his talk and request Rajesh to present a memento.
Present, wow.
A small memento from my CPS.
Thank you. Okay, thank you. I actually have a coffee mug collection, so we'll be very happy to. Okay.
We'll have questions at the end of the talk.
Okay, okay, thank you all for coming. I'm going to try to give a very general talk about what we all care about, which is why has it been so hard to actually come up with a cure for cancer? Now, 30 years ago, more than 30 years ago, the President of the United States, Richard Nixon, announced a war on cancer. A war on cancer meant that all of a sudden a large amount of research money was made available to the United States biomedical community to try to attack cancer, with the idea that drug therapies, other methods, radiation, would very quickly be able to come up with a cure for this disease. Now, actually, this, if you look historically, this had a very positive impact on the biomedical community in the United States. One of the strange facts about this war on cancer is this was done in an age when people thought that the cause of many cancers were viruses. It's true that there are viruses that cause cancers, but it's probably not the major, or certainly not one of the major causes. But because of that, a lot of money was spent on viruses and how you detect them, how you measure their features, and that turned out to be essential for actually treating HIV. When it first arose, it was easy to discover how it worked and what it was, and eventually how to treat it. But in fact, the effects on the cancer problem were much more limited than that. And here on the bottom, you can see sort of the current statistics, which is that the worldwide mortality per year in cancer is sort of on the order of 10 million people, which is a lot of people. In the US, it's getting very close to being the second most common cause of mortality past heart attacks and heart disease in general. And the reason for that is partially because we've actually been rather successful in creating methods to treat cardiac disease, coronary disease, other related strokes, other related things, whereas we've been less successful in being able to treat cancer. Not unsuccessful. If you look at this picture on the bottom, you notice a very large discrepancy between different types of cancer. There's, of course, also the incidence rate, but even given the incidence rate, there are certain cancers which we've been relatively successful at treating, other ones we've been relatively unsuccessful in treating, and we don't completely understand yet why that's the case. Okay, So let's move on to the science part. So I'm going to talk about sort of two parts. One is I've titled Know the Enemy, which is, can we understand something about cancer cells, why they're so effective at killing us, and why we've been relatively unsuccessful at killing them. And the parts I will talk about, we've heard a lot about that today at our specialized talks about drug resistance. I'll talk a little bit about that. I'll talk a little bit more about metastasis. Metastasis, of course, is the process by which cancer cells leave their original source, their primary tumor location, and spread throughout the body, taking up residence in other parts of the body, growing new tumors there. And roughly speaking, metastasis is really the part of the cancer cycle that leads most often to patient death. It's mainly untreatable, and we don't really know what to do about it, and you can delay death for a couple of years. But once your cancer metastasizes, in most cases, you have a pretty limited lifespan afterwards. And then I'll talk a little bit, because this is a workshop about how theoretical ideas and how ideas coming from physical science can help. I'll talk a little bit about how computation and theory at least has helped try to understand some of this. Then I'll turn to a second part of the talk, which is I'll call Know Thyself, which is, of course, a combination of the famous statement that if you want to win a war, you need to both know yourself and know your enemy. Well, the second part is knowing ourselves. In this case, ourselves meaning what we have in our bodies, which are the methodologies for trying to attack cancer cells and kill them, namely our immune system. So I'll talk a little bit about our immune system. Why is our immune system less successful at attacking cancer than attacking viruses or bacteria, although as we've discovered in the last few years, it's not always that successful in attacking viruses. And then we'll talk a little bit about immunotherapy, and will that be the magic bullet? What's stopping it from really finally giving us the answer to Richard Nixon's request of 30 years ago? Okay. So what is cancer? Well, what is this disease we call cancer? And it's a very strange disease because it actually is a whole combination of different diseases in different organs, in different parts of the body that arise from different causes. And why do we even give it the same name? It's sort of a whole range of different maladies. And the answer is because there is something in common about this disease. What's common about this disease among all the different causes and all the different locations is that it's really a breakdown in the cooperation of our cells. Our cells, have learned to cooperate with each other to make the community of cells that we call the organism, i.e. our bodies. And in order to do that, they've had to give up various freedoms that they have. They can't just grow as they wish. They can't just move from one part of the body to another as they wish. They need to have a fixed role. They need to stay in place. They need to grow when growth is essential, stop growing when growth is deleterious. And those results, those constraints on the individual cell are not left up to the individual, well, they're left up to the individual cell's network, but they're essentially forced upon the cell by its coupling to all its neighbors. So it's as if the cell lives in some type of benevolent dictatorship, where for the good of everybody, they're being told what to do and when to do it, and they have to listen because they have structures built into them that make them listen. Those structures are called biological networks. They're networks in the same sense that an electrical system or a circuit is a network. It's a set of interconnected components that act to take in information, have some input nodes, do some processing, have some output nodes, and essentially define what cell behaviors will exist in response to various perturbations, various changes in the cell's environment. Information coming from the external non-living parts of the environment, and of course, information coming from all the other cells in their vicinity. And so what cancer is basically a breakdown of that benevolent dictatorship. It's a breakdown of those parts of the network that tell the cell what to do in a way that is good for the whole, but not necessarily best for the individual cell, thought of as a completely selfish player. And this has been stated in a number of ways by a number of people, and the basic idea here is that cancer, therefore, in this way of thinking about it, corresponds to the dysregulation of many of the regulatory control systems of a cell well, almost always started off by some type of genetic mutations that break some of those bonds here. And then you can actually see how this then evolves into something where the cells are acting in a much more selfish way. Now, here is a picture of the cells acting in the way they're supposed to be acting. Just an example of that type of piece of the network. There's a particular protein, which is a particular node in those networks. It's called P53. P53 has it's not a very exciting name, but P53 is actually the most common gene that's mutated in cancers. In other words, if you go and look at all types of cancers from all across the body, from all these different causes, you will discover that in many, many of them, P53 has somehow had its functions disabled. What are those functions of P53? Well, here is a diagram of what those look like. I don't want to go through this in detail, but essentially, P53 is the object that says, if things are going wrong, if things don't look right in my vicinity, if I have some type of uncontrollable stress, those things can be some type of malfunction in my machinery, that's ribosome malfunction up on top here, some type of radiation that's come in and has mutated my DNA. Well, if all those things come in and I maybe try a little bit to fix it, if that doesn't work, my cell is supposed to commit suicide. Committing suicide is not the best plan of action for the cell, but very often may be the best plan of action for the body. Well, if P53 is mutated and those actions come in, let's say ionizing radiation, let's say residual chemicals from cigarette smoke, whatever it is, and P53 becomes inactivated, all of a sudden the cell is ignoring the instruction to commit suicide for the good of the whole, and it's going to try to do something to counteract that particular stress. It's going to try to mutate, it's going to try to evolve, it's going to try to adapt, and in the course of that, it's going to start a cascade that can lead to cancer. That's the way to think about what's going on here. So, in terms of this, whoops, that's the wrong thing to press on. I always do that. That's weird. Okay, hold on a second. Okay, so if you think about it, we do know of cells that behave in that way already. We usually think of them as, nuisances to us, but we can actually learn something from them. It turns out that if you look in the bacterial world, the microbial world and bacteria are very much acting the way that cancer cells start to act when they disable all these regulatory things. So, just as a matter of historical note, my foray into cancer research occurred about, let's say, a little more than 10 years ago by now. We decided that this would be something good to try to tackle given advances in the knowledge of biological physics and computational methods. And had a partner in doing this, Eshel Ben Jacob, who has unfortunately passed away. And our first attempt at understanding what was going on in this field was to make for ourselves a sort of conceptual roadmap as to how we were going to think about the disease and think about how one should begin to study it. And that was then summarized in this paper about how bacterial survival strategies suggest rethinking how cancer works. And so that was the start of my personal journey into trying to understand some of these questions. Now, that way of thinking leads one to begin to understand two separate phenomena that I want to describe. I said, one, we had many talks, many excellent technical talks about this today. Let me describe it at a little less technical level. The first question is drug resistance. So one of the problems with metastatic disease, with cancers that spread throughout the body, is that those cells somehow become very non-responsive to all the drugs that we spend millions and billions of dollars to develop. Somehow the cancer cells have learned to adapt their way out of being sensitive to those drugs. Now, it turns out that bacteria do the same thing. One of the problems we have in the world now is the bacteria are very efficient at beating our drug industry and coming up with ways of evolving antibiotic resistance. Now, those processes that give rise to resistance can happen in a number of ways. Originally, it was thought that almost all the time that happened by mutations, and mutations are a strategy which, it first of all is greatly benefited by having more of you. The faster you grow, the faster you reproduce, the more mutation attempts you'll have, the more chance you'll have of coming up with a mutation which will defeat the thing which is causing you stress. So the bacteria, for the simplest strategy, is just grow as fast as you can, have billions, billions of bacteria, and maybe a few of them will luckily figure out by accident what to do, what type of mutation will be enough to defeat the antibiotic that you're using to kill them. Now that's a very powerful method when you have billions of individuals basically trying to do that. And in fact, that is the cause of many of our problems with treating bacteria these days, where you have multi-drug resistant tuberculosis and other diseases, which are a real challenge for ongoing medicine in our century. There's, of course, another possibility. The other possibility is it's not really a mutation. A mutation means a change in the actual DNA of the cell. The other possibility is that what it's called a cancer persistence or drug persistence, where the cell has figured out how to adapt its behavior to not be sensitive to the drug. So for example, if your drug is killing cells because they reproduce in some way, then you can reproduce less often. You can go into some more static state and become a persister, a drug-tolerant persister state, and not reproduce. If your drug is figuring out how to destroy your membrane, you can basically pump that drug out. There are all sorts of ways in which bacteria, if they're able to experiment, if they're able to explore different possibilities for their behavior, will eventually defeat drugs. Well, cancer cells are not supposed to be doing that. They're supposed to be controlled in their growth, for sure. They're supposed to be controlled also in this exploration. And the answer is, well, that's what the disease has disabled. The disease has disabled the controls. They now behave like the bacterial cells, and they're able to do this exploration and figure out how to defeat these drugs. Now, there are lots of, there are tons of work that's on here. I wanted to give you just an example of what research looks like in this area. Not meant to worry about the details. This is from a particular paper, already by now a decade old or more. It's a case of lung cancer. And the idea is, if you take lung cancer cells up here, this is percentage of cells that live. If you take lung cancer cells in the lab, and you dump some chemical on them, that's a targeted therapy that tries to kill the cells, what you'll discover is the targeted therapy is pretty good. It will kill 98% of the cells. The problem with killing 98% of the cells is that the 2% that's left over, given enough time, will figure out how to regrow, and they'll start exponentially growing again, and pretty soon you won't have 2% of your cells left, you'll have 100% of your cells left. So the exponential growth of residual cells is basically, it gives you almost an impossible task of killing every single last one of the cells there, and that just doesn't happen. There are cells that are able to survive, are able to adapt, and here you can see what happens if you sort of track over time. So here is untreated, you see a whole bunch of cells here, then you dump the chemical on, you see that almost all the cells have been cleared off, you see a bunch of leftover dots there, those are cells that are sort of hanging on, and then you wait a month, and all of a sudden, those little dots that were hanging on, some of them have started growing again, repopulating the microscope side. Well, if you think about this for a patient, these are the cells that are then repopulating the tumor that you thought you wiped out by giving your therapy. So the consequence for a patient looks like this. You have a picture which is sort of two classical ways in which the cancer community can sort of show this phenomena in action. The way number one up here is called a Kaplan-Meier plot. It's basically a plot of your survival given that you have a tumor with and without a drug. So if you don't give a drug, that's the sort of white line over here. If you give this particular drug, then you've sort of pushed your survival by a couple of months. Now, fortunately or unfortunately, if you get a drug that can push survival on average by a couple of months, you're a success in the biotech world, because you can sell the drug, you can treat some patients. Most of the time, they'll be happy for getting a few extra months of life. You can make lots of money, but it doesn't really solve the problem. It's just really delayed the inevitable for a few months. And what's the problem? The problem is exactly what I said before. You give the drug, and here is a sort of illustration of what happens. This is called a waterfall plot. And these are individual patients. And what's plotted here is how much was the maximum reduction in the size of the tumor during the therapy. So the way this therapy works in the clinic is you give the therapy, you measure via some imaging technique what's happening to the tumor, so hopefully the tumor is shrinking. There are a few people for which the tumor doesn't even shrink at all. Okay, that's just a small number. Most of the people have some degree of tumor shrinkage, but then in some cases it goes down to 40%, in some cases it goes down to 60%, but most of the people go down to some level and then have a rebound. So you have a minimum disease, and then the tumor starts growing again. Why did it start growing again? Because some of the cells that you didn't succeed in killing have regained the ability to grow, and now it's repopulating the tumor. And of course, those cells that are repopulating are now immune to this drug, so giving the drug again is not going to help. And so you basically have gotten some amount of time when you've reduced the tumor, and eventually it just regrows back to where it was before, and you're sort of back to where you started. In the clinic, it means you get a different drug, and eventually they run out of drugs and you run out of time. Of course, there are some lucky patients down here that have sort of 100% remission. Even 100% remission doesn't really guarantee that you're free of the disease, because imaging methods are only so good. You can't detect individual cells scattered in random places throughout your body. And so even if you get what's called clinically, full remission, it doesn't mean that there's no cancer cells in your body. It means that they're below the resolution of the imaging technique. And in many of those cases, 5 years later, 10 years later, whatever, you go back to the clinic and discover that you have metastatic disease, because those cells that weren't even detected in the scan are really there and can really start growing again, given enough time to adapt to their circumstance. Okay. Now, let me switch to the second part of what adaptation manages to do for cancers. And that has to do with not drug resistance, but the idea of metastasis. So metastasis, as here is a schematic diagram from the lab of Bob Weinberg, who, by the way, I like to say, that he really did write the book on cancer. He wrote the leading textbook on cancer biology. He had this nice cartoon. I really love the cartoons that the cancer biology community makes. It gives you great insight into how they're actually thinking about problems. So here is his picture of how metastasis is supposed to work. His picture was the following. Here, in this green here, is you have a primary tumor. And the primary tumor consists of cells that are called epithelial cells. Now, epithelial cells are the cells that make up most of the organs of our body. So think about the breast, think about the pancreas, think about the lung. They're made up of cells that stick to each other, form layers, form circular or spherical geometries, form ducts, have some geometry which is made up by these cells sticking to each other and creating some functional geometry that lets the organ do what it's trying to do. It's in the air, in the lung, it's trying to create a surface that will exchange air with the outside world. In the breast or the salivary gland, it's trying to be able to secrete things. In the pancreas, it's undergoing various chemical transformations of things passing in the kidney, passing through. So those are epithelial tissues. Now, epithelial cells stick to each other and aren't going anywhere. They're normal body tissues who have been instructed not to divide and to not go anywhere. Now, when you start having the cancer, the first thing that breaks is the constraint to not divide. So you get a primary tumor. The primary tumor is a breakdown in the growth control. All of a sudden, you get a mass that appears in that part of your body when it shouldn't be there. And it's not good for you, but we can treat it. We have therapies such as surgery, such as radiation, that if the mass remains localized in one place, you can do a pretty good job of getting rid of almost all those cancer cells. But the problem is that just as the growth control can break down, the control over staying in one place can break down, and then cells can start to pick up new properties, which are here called mesenchymal properties. The terminology doesn't matter for those of you who are not in the field. It basically just means cells that move. And now these cells can leave this green region. They travel, they make it through the blood system, they travel, they go to other parts of the body. So here is going to the lungs, and then they start growing a new tumor in this place over here. Now, I think you have to understand that this looks simple on the cartoon, but is really, if you think about it, a very hard task for a cell to do. all of a sudden the cell has to pick up, decide not to stick along with any of its neighbors, any of its very comfortable surroundings and geometry, both physically and chemically, travel long distances. I mean, a cell is, 10 to 20 microns. It's got to travel across your body to a new organ. That's a very far distance. It's got to land it's got to make it through the blood system, which is sort of like surfing down Niagara Falls or something for a cell. It's really very turbulent flow that can disturb the cell in many ways. And then finally, the last part is they have to emerge in some totally foreign country, and they have to adapt to all sorts of new languages, customs, whatever. Now, you probably have much more experience in something like that than I do, at least the ones of you who have picked up and spent some years in a foreign country. But it's not easy, I mean, it's all of a sudden everything that you know how to do and you knew how to order coffee, and you knew which way to walk down the street because the car was coming on that side, all of a sudden is different. And if you don't adapt, you die. And in fact, the vast majority of cells that make it to this other part of the body die. However, some of them don't. And again, you were at the victim of exponential growth. If only a few of them survive, it's already curtains, because those few of them can seed a new tumor, which can grow exponentially, and now you have tumor spread throughout these other parts of the body. But you can ask the question from a scientific point of view, what is it that gives the cells the adaptability to actually do all these things? So this brings us to how ideas from physics or computation or engineering, whichever subject you want to think about, are able to at least partially contribute to understanding something like this. So in order to undergo this transition from the primary tumor to this cell that's motile, that can move along, survive, get somewhere else, in order to do that, it has to change its fate. It has to change its nature. And how do cells change their nature? What do we even mean by cells change their nature? Well, cells, you can think of as being some type of rudimentary information processing system. An information processing system can take inputs from the outside, and it can make decisions about how they're going to behave in response to that information coming from the outside. And the way they do it is the same way, some electronic circuit that's acting as a sensor with then some type of output from the electronics that's determined by the sensor. It's done by some type of circuit. Here is now an example of one of those circuits. Before I showed you a very abstract picture. Here, this is a sort of schematic of the circuit. This is due to Rika Albert. And it sort of shows in some form what are all the pieces of that sort of computational engine that's taking the input. So the way to read this circuit is inputs come in on the top. These things on the top are all things that get information from the outside. They're all proteins that sit on the surface of the cell, detecting molecules, detecting forces that are impinging on the cell. And then this is stuff that sort of, takes the signal and integrates it with other information, like the state of the cell, like, is the cell hungry, is it not? So sort of, takes internal information and mixes it with external information. Finally, you come down to the bottom, and at the bottom, you have a module, which essentially is the output module, makes a decision about what the cell is going to be. So this is a network whose job is it to take inputs from the outside, make measurements of the outside, figure out something about the internal state of the cell, and decide, is the cell going to stay as an epithelial cell, as it's supposed to, or is it going to make a transition to a mesenchymal cell? Now you might ask, why would a cell have a machinery to do this when it's really bad for a cancer cell to make this decision? And the answer is because cancer is not the only reason for cells to exist. The reasons that we have developmental processes that build us from a single fertilized cell, and those developmental processes require cells move throughout the embryo. It turns out that the ability to change from stationary cells to motile cells is a key part of developmental biology. That some cells start out in one part of our embryo, they need to spread out to create blood systems, to create skin, to create muscles, and those cells are doing what they're supposed to do. They're programmed to do that. This is the circuit that programs them to do that, and this circuit has been hijacked, it's one language people like to use, by cancer cells to do something which is now harmful. So it's not a circuit that's been invented by cancer cells. Cancer cells have only been around for a few years in our body. They can't evolve an entire new thing like this. This is a billion years worth of evolution in this picture, but there you can make small modifications to existing circuitry. In particular, they can remove some of the requirements. So this circuit, which is buried in the midst here, will tell an epithelial cell that if it doesn't get the right type of signal, then not to do this. On the other hand, the cancer cell can break that link, can ignore that and say, I'm doing it anyway. And that's the problem. Okay. So now you can analyze this, and we analyze this as work that we started to do about 10 years ago, and this is part of Mohit's thesis. So I figured I would show this, and so we showed that actually when you did this, the cell, so we analyzed that piece of the circuit. I won't talk about the details, but what turned out to be the case is that cells sort of had three choices in their life. People in the biology world, in that cartoon, it looked like the cells had two choices. They could either be epithelial or they can be mesenchymal. And you could either be static and not moving, or they could be individual and moving. And it turned out that that was a oversimplification. That oversimplification was discovered by actually trying to understand the circuitry. And the oversimplification was that it ignored the possibility that cells could reach a compromise. The compromise is they could have what we called a partial EMT state or a hybrid state. The hybrid state was something that had properties that were both partially epithelial, cells still stuck to each other, but they also were partially mesenchymal, which means now they had the ability to move. And what we're able to show, working with cancer biologists over a number of years, is that when these cells would leave a tumor, instead of leaving as individuals, because the previous transition to purely mesenchymal states cancels all their cell-cell stickiness, these cells, if you only undergo this partial EMT state, actually the cells leave as some type of cluster of cells. They leave as a stream of cells which breaks up into clusters. Okay, great. So we did that. We checked this in other models. I won't talk about that in detail. But it turns out that the fact that they left as clusters had a very, very significant role to play in our changing the thinking of what was going on. Now, what was the role? The role was that I said before, the hardest thing to understand is how does the cell survive all these challenges? How does it actually go from this epithelial cell all the way to another organ, going through all those challenges? How does it survive? And our answer was, of course, well, the cell must somehow be very adaptive. It must be able to figure out what it needs to do to survive. And of course, many of them will die. Most of them will die. But some of them will be adaptive enough that they will be able to survive and eventually flourish in a new environment. And it turned out that when we did the calculation of figuring out how adaptive those different cell states were, the E state, the hybrid state versus the mesenchymal state, it turned out that the hybrid state had all the properties one would expect for a very adaptive cell. So the hybrid state is not just, oh, well, it's a third choice. It turns out it's the most important choice. Cells that are hybrid are the ones that we felt could survive. Now, we were able to check this in a number of in vitro experiments. Now, in the cancer world, you have to start with in vitro experiments because they're relatively inexpensive. You have cells growing in a dish. You know what you're doing. You can handle them. But you can't get believed for anything until you start doing experiments in real animals. It's hard to do experiments in people, so usually the animal du jour is the mouse. So people started eventually at least partially believing what we were saying, and they set out to do experiments in mice. Here is one of the first ones that really sort of changed everybody's thinking. I'll just explain this experiment. They took a tumor in a mouse. And this is, so this is a paper that appeared in Nature. This is SCC, which is some type of skin cancer. And they took a tumor in a mouse, and they removed the tumor from the mouse. And then they said, well, gee, based on all this work that people had done, classifying these different states and understanding what are the different ways in which this network is creating, genes in these different states, we can try to classify all the cells in our tumor that we just extracted from the mouse we can try to classify those cells in terms of their EMT status. Now, it turned out that, when they did this in their own way, they decided that, well, E was a unique state, right? M was a unique state, and then there were certain gradations of partial EMT. There was they came up with 4 gradations of partial EMT. But the real experiment was they then took the cells, they sorted them into these different piles, and then they took the different piles and injected them into new mice. And the idea was they wanted to see which of those cells were actually able to grow new tumors. And the answer, which is given down here, is that the cells in the intermediate epithelial and mesenchymal phenotype, rather than the ones that underwent complete EMT, are the most metastatic populations. So they directly verified what had been calculated based on the circuits, based on the analysis, and then based on working directly on in vitro experiments. This was the first example of this in an actual in vivo system. This, I think, broke the tide. Now, if you go to most cancer biologists, they at least acknowledge that this is a strong possibility, that cells that are in these partial EMT states are really the ones that are the most adaptable, the most capable of metastasis. And of course, eventually it gives us some idea that those are the ones you'd have to target if you want to really get rid of the metastatic possibility. These may be a small percentage of the entire tumor, but these are the ones you have to figure out how to kill in great numbers, because you can't leave any of them to survive if you want to actually get rid of metastasis. One side note, just a sociological side note, which I always like. It's always fun coming from one field and trying to work in another field. It's sort of like metastasis in a way. So here you see, something which I always love this. This is from the News and Views article based on this paper. So it was exciting to see that in contrast to what one would expect, the tumor cells, et cetera, et cetera. Now, in contrast to what one would expect is a very interesting statement, because they actually cited our paper and other papers which predicted this. So what does it mean to in contrast to what one would expect if it was predicted and, in fact, they cite the prediction, which was very nice of them to do that. And the answer, you can come up with your own theory. I have several, and you can try them all out. Number one is that they cited our paper, they read it and didn't understand the prediction. That's number one. Number two is they cited our paper, they read it, they did understand that it didn't believe the prediction. Okay. Number three is that they actually did expect this, which is why they did the experiment, but in order to publish it in Nature, you had to claim that they didn't expect it. So that, to me, that's probably the most likely explanation, but you never know. I'm not asked the author of the paper, the first author of the paper, this question, but maybe I should one of these years. Okay, so, but the point was that the ability to sit down, think logically about the network that was controlling these fates, work with cancer biologists to then do very carefully engineered experiments to test this idea, really did have a contribution to changing what people thought about the metastatic process. So I think that's quite an interesting ability. So here is the lesson so far of the sort of first 2/3 of what I wanted to say. So cancer cells are much freer to adapt to changing conditions because of the fact that that is what cancer is. It's the loosening of these network-driven constraints. This enables cells to do many things that we don't like, defeat drug therapy, establish metastatic growths, and therefore we need a counter strategy that's going to be as equally as agile as the cells are if we're going to hope to really cure cancer or really make inroads in many cases. So in some cases, regular drug therapy will be enough. We'll be able to kill enough of the cells that this won't really happen, or maybe it will happen 20 years later. There's a famous statement about prostate cancer for men, which is that all men die with prostate cancer, very few men die of prostate cancer. That if you look in the prostate of everybody my age, you find cancer cells. It's just that by the time that they will grow and form something and metastasize, for most people, we will die of something else. So that is one way to look at it. We can do that. We can just delay things and hope for the best. Or there's another possibility, which is to use the immune system. So in the last 20 minutes, I want to talk about last 20, 30 minutes, talk about the human immune system as the other side of the coin, the know thyself side of the coin. Okay, so how does our immune system detect things like cancer cells? What's the logic? So the immune system is an incredibly complicated object. It has many, many layers, going from physical barriers like our skin that prevents things from coming in, to then cells that detect bad things that come in and try to interdict them. Eventually you reach the adaptive immune system, which of course has gotten a lot of publicity for how it deals with things like COVID-19. And the adaptive immune system is really the part that is trying to do something about cancer cells. Now, the part of the adaptive immune system that's relevant is the part that is called the T cell arm of the cancer cells. The way T cells work is that they look for cells that have somehow gone wrong. So they're sort of in the business of interrogating other cells and figuring out if there's something wrong with that other cell. Now, the something wrong is very often that other cell has been infected by a virus. And therefore, what they're really looking for most of the time are these viral infected cells. If they detect that the cell has been infected by a virus, their instruction is to kill that cell. Now, how do they know how do they look on the inside of something if they're stuck on the outside, right? The T cell it's not entering into the other cell and taking a survey. How does it figure out? So the answer, I think, is sort of, I remember a long time ago, whoops, doesn't go to the next one here. Back. Let's see. Why doesn't it let me show this? Let's click on that maybe. Yeah, no, but I wanted to go to yeah, oh, fine. So it's sort of like this amazing thing I learned decades ago in Japan. So in Japan, you have trucks driving down the highway, and there's these series of lights on top of the truck. It's where that arrow is. There's sort of a series of 3 lights. And the truck has to display on the outside of itself what speed it's going at by this light system. So between, 10 and 20 miles an hour, one light goes on. Between 20 and 40, another light goes on. Over 40, 3 lights goes on. Now, why do they do this? Because it saves the police effort. They don't have to have a radar gun and sit there and detect what's going on. They can just see these lights, and they can see how fast the truck is going, and they can decide, is there a problem, is there not a problem, and they can write a ticket if too many lights are on, given the roadway. So the trucks are displaying on their outside something about their behavior that then is easily seen by some survey system. Now, that's what T cells do. T cells survey the surface of the cells, and our cells have a system which is the analogous system of this light system. We have proteins that take pieces of our proteins from the inside, display them on the surface for the T cells to see, and if the T cell sees that it sees something it doesn't like, then it turns, it does a number of things. It first of all says something is wrong, so it sends instructions to other T cells to come by. It then divides to make lots more of itself, and eventually then it kills the cells that have the bad signal. Okay, now, we're intelligent too, just like the Japanese. So you might say, well, what happens if a truck driver doesn't do this, or lets this thing fall into accidental disrepair? And the answer is, if you don't have the lights, you automatically get a ticket, obviously. Well, our cells have the same thing. If you disable this presentation mode, if a cancer cell decides, if a cancer cell breaks the signaling mechanism that displays these proteins on their outside, we have other cells, they're called natural killer cells, that come along and kill those cells. So you have to show this, otherwise you'll be killed, and then you're killed if you show the wrong thing. That's the way to think about the system. Now, so here's a picture. Whoa, what happened? Oh, okay, I pressed the wrong button. Okay, sorry about that. I know I usually don't use these things, but it's too far over there. So here's a picture of what this looks like. These are T cells interrogating a cell by sticking to it and trying to measure what those proteins are on the surface. This is a more molecular view of what's going on. This is the T cell. It has some protein on its surface called a receptor, T cell receptor. Here is a cell such as the tumor cell or maybe some other cell. It has this machinery that puts a piece of a protein right at that spot, and then these things decide if they can bind to each other, and binding to each other constitutes a detection of this cell by the T cell over here. That's the basic mechanism. Now, you might ask, you should ask, well, okay, every cell is displaying something. How do I set up a system like this such that it's able to detect bad things, foreign things, but not able to detect ourselves, right? You want to have a way of creating such a system that if a cell has gone bad, if it's displaying a protein that, for example, came from the virus, a piece of a protein that came from a virus, let's say a piece of the spike protein from COVID-19, that cell should be killed. On the other hand, if it's displaying a piece of one of our proteins that's supposed to be there, it's supposed to ignore that. So how do you set up a system like that? So there's, of course, a rather interesting procedure. We generate a huge number of different T cells with different receptors, and they're going to detect basically almost everything, right? It's a coverage of, you generate about 10 to the 7th, 10 to the 8th different receptors. So a whole, slew of different receptors that go around testing these things. And so you can detect pretty much anything, any sequence. And now what you do is you go send these T cells to training school, and you say, well, I take my T cells. The T cells are called T cells because they go through a part of the body called the thymus. The thymus has the property that it engages in negative selection. The thymus is full of peptides, small protein segments that come from our cells. The T cell passes through. If it binds to one of those things that comes from our self, then it says, oh, we don't want to release this T cell into the blood system. It's going to attack one of our own cells. The T cell is told to commit suicide. Again, one of those, cooperative things that the body is able to set up by having these regulatory links. If it doesn't do that, if it manages to pass through the thymus, and not bind anything, it's assumed that it's not going to bind any common protein in our body, and therefore it's free to be released, and therefore if it binds to something outside of the thymus, it's thought that that thing is most likely going to be some foreign, infected cell, and it's going to kill it. So this is a very, very nice system, and you can ask, is this system good enough to detect tumors versus our own cells? What's the problem? The problem is that this system probably evolved to detect viruses or maybe bacteria that enter into cells. Some bacteria enter into our own cells. Those proteins that come from viruses have, only statistically have nothing to do with our proteins. They're not similar to us. Of course, there are only 20 amino acids. There's only a limited number of chains of 15 amino acids you can make, but there's a large number compared to how many proteins we have. The chance of there being a very close connection between a piece of 15 amino acids taken from a viral protein versus something taken from one of our own proteins is small enough that, you won't have any problem telling the difference. It's really a huge difference between viral pieces and our own pieces. But a tumor is not a virus. A tumor is our own cell that has some small number of mutations. So if you look at the proteins of the tumor, most of them are the same as a normal cell. A few of them are mutated, but only slightly. They only slightly differ from our own proteins. So you have to have enough resolution to figure out the difference between a protein taken from one of our cells, let's say that's of length, 10 to 15 amino acids long, from something that is the same length long, but has one amino acid change in it. That's the resolution you need if you want to tell the difference between a tumor cell and our cell. That's called a tumor neoantigen. It's an antigen because it has one mutation in that. Now, it turns out that that's possible. And it's an amazing engineering feat to make this possible. This is really bad here. There we go, okay. Now, one way we know this is possible, by the way, is this nice experimental fact, which is you can figure out which types of tumors are most susceptible to being attacked by the immune system. Now, it turns out, if you think about it, that tumors are not equally filled with mutations. You have tumors like melanoma, where you go out in the sun every day, and especially if you have skin like mine, as opposed to most of yours, you are exposed to UV radiation, which damages your skin, and you pick up mutations, and that's eventually the cause of skin cancers like melanoma. Other diseases like lung cancer from smoking, where you have chemicals which create large numbers of mutations, create tumors that are highly mutagenized compared to the normal cells. So those tumors have many more opportunities for detection than ones that have small numbers of mutations. It turns out that if you make a graph, this was done in the cancer biology community, if you make a graph of tumors and list them according to how mutagenic they are, how many mutations they have compared to normal tissue, that correlates pretty well with how well the immune system is able to attack them. So we can see that the immune system is relying on these mutations to create slightly changed peptides to be detected by the immune system. There are other arguments. In fact, there are cases where you can directly look for these things. There's another argument, which is a more speculative idea that we proposed and other people proposed a couple of years ago, which is it's well known that immune systems decay with age. Many things decay with age, but one of the things is immune systems. And in fact, it turns out that even worse for me, that men's immune systems decay faster than women's immune systems. Now, if one of the things that's preventing tumors from growing in your body typically is your immune system, then you would expect that as you get older, your chance of incidence of cancer, all else being equal, would go up because your immune system is getting less and less effective at battling anything. So it turns out that that's, of course, true, that there is a very large increase in cancer incidence with age. It's usually attributed to other factors, but nonetheless, you can fit the data with a theory that just says that the only thing that's changed is your immune system, and then you can get the same data. You can explain why the incidence curve for men is sharper than it is for women. You can explain why people who get kidney transplants, who then go on immune suppressive drugs, tend to pick up cancers more often than people who are not on immune suppressive drugs. So you can make a reasonable argument that the immune system can, in fact, attack cancers just from a more phenomenological point of view. But you don't need the phenomenological point of view. You can also directly show that there will be T cells that can attack cancer cells in the body, and people have shown those things directly. Okay, now, this is really bad. I keep pressing the wrong button. Sort of like when I try to type text into my phone. Now, how can this work? So when I do problems like this, I always find that the biology literature is just so immense that if I just go and sit down and start reading biology papers, I get lost. Now, of course, there are other people who can do that and don't get lost, but I just, after about the second paper, it's like, I just can't take it anymore. So I always need some conceptual framework that I can sort of create for myself that will allow me to sort of read papers in a much more efficient, of course, maybe I'll be wrong about my conceptual framework, but at least I can sort of say, oh, this paper has something to say about how I'm thinking about the problem. Either it's saying I'm on the right track or on the wrong track. But then most of the papers will have nothing to do with how I'm thinking about the problem, and I'll just say, oh, I'll put that in this pile and read another day, and another day doesn't come. So we wanted to create a conceptual idea as to how this could work. And in this case, the we was myself and a different student of mine, Jason George, who actually will be here next week giving talks. He's now a faculty member who continues to work on the immune system. And the logic is the following, and I always think of this analogy, which I think helps understand what this graph is trying to convey. So think about, let's say you're having a party. You're a graduate student, you want to have a party, but you don't want to invite any physics graduate students because you think that they're just downers. They're not going to be very good for your party. But you don't know who's a physics graduate student and who's not a physics graduate student. So what are you going to do? Well, you have two choices that roughly correspond to two different ways of thinking about the problem. The first choice is to try to find features of physics graduate students. So what will you do? You will go and make a study of all the features of the physics graduate students, how they wear their hair, how many wear glasses, all these things, and create a feature map and create a filter, and anybody who comes in, you'll be able to detect immediately if they're a physics graduate student because your features will be very good. And that's how you're going to decide to not invite them to your party. The problem is you're probably going to rule out lots of chemistry students, lots of engineering students also, because enough of them are going to look like physics students for that to fail. So you'll make a mistake, and you'll make one specific type of mistake. You'll make a mistake of throwing out too many people. You'll make you'll make mistakes of false positives. You'll think those people are really physics students when they're not. Okay, the other possibility is you go and sit in the physics building, and you take pictures every day, and you memorize all those pictures. And then when it comes time to issue your invitations, you compare the list with all your pictures, and you say, oh, this is a physics student, this is a physics student, this is not. I just invite them. So I just memorize who all the physics students are. Of course, now I have the other problem, is now if somebody was not available that day, they were sick or they were traveling or whatever, then they didn't happen to show up in the 2 weeks you had to do the survey, and now you invite them even though you shouldn't have invited them. So those are the two choices you have, a feature detection choice or a memorization choice. If you do a feature detection, you're gonna get a curve which looks sort of like this. You're gonna cover space of these of all the students, or in this case, all the peptide fragments, with these yellow markers. You're gonna say, oh, everybody in this region is something I want to get rid of. And so now when you go through this negative selection and you throw out things that are similar to our own peptide, you're going to throw out anything in the vicinity of those peptides. And you'll throw out tumors cell also. So you'll get something that looks like this. The detection will have a slow increase away from the baseline. By the time you're only one step away, you're not going to have very good detection capability. In this case, you have very good detection capability, because now, as long as you didn't memorize that particular sequence, if it's one base difference, it's not on your list of memorization, so now you can detect it. Okay, so our argument was that if the system is going to work as advertised, it must somehow be similar to this rather than to that. It must be that you're not detecting features, you're really just memorizing a list of things that you don't want to have T cells attack. And if it's not on the list, then those T cells are fine to go. Now, how do you test an idea like this? So there's 3 ways to test the idea. One way is to say, is this even possible? Can you make some type of model that will give rise to this behavior? So the answer is, we sat down. In this case, I said Jason and I sat down, and we made a model. It's called a random energy model. It's a type of model that was used extensively in the world of protein folding to understand what was possible, what was not possible. And this model, you can go through, you can calculate things analytically, which is great. Every once in a while, it's nice to do an analytic calculation. And you can show that in this type of model, you can actually get that behavior. The penalty for being a tumor neoantigen as opposed to being a completely orthogonal virus is only about a factor of 2 or so. So you're really not taking a very large hit by getting rid of things that are regular peptides. You still have enough residual capability that you can detect tumor neoantigens. So it can work. It doesn't mean it does work, it means it can work. Okay, that's great. The second way is to rely on your friends. So you have friends out there who are looking at the same problem, and luckily for you, they're looking at it in a slightly different way than you're looking at it. So this group, rather well-known physics group, by the way, looked at the problem the other way. They said, well, let's say you wanted to make features. Are there really features out there? So are there really things that you could say, this is a human, real, honest-to-God peptide from a normal cell, and this is really a tumor neoantigen? Is there something that tells you the difference between the two, such that the first methodology of creating features could actually work? So they investigated this in great detail, looking at all the sequences, all the structures of all these different things that were known, and they concluded that it just doesn't work. The distributions of self and non-self peptides are nearly identical. They could not tell they had no leverage to actually create these features. Discrimination is only possible because you're drawing a finite sample and the immune system can target the spaces. This is another a long way of saying you memorize the answer rather than try to generalize it. And then in conventional learning problems, this is called overfitting, leads to disaster. So you would never do this if you were doing machine learning, but we're not doing machine learning. We're trying to solve a different problem. Now, the last point about the way to argue that this is at least possible is to say that this predicts something about the experimental data that would otherwise be maybe a little bit more surprising. So the other thing is that there have been papers in the immunology literature around the same time, a little bit before, which shows that, in fact, you make mistakes of exactly the type that you would have thought you make when you do this memorization task. Because the T cell has only a finite amount of time to sit there in the thymus sampling self-peptides. So it will miss some. It just won't happen to hit that particular cell with that particular display, and so there will be mistakes of a certain type that's indicative of the selection criteria. Now, those mistakes have to be fixed, otherwise we would all have autoimmune disease. So it means there have to be things called peripheral tolerance, which means there have to be higher levels of checking. It's not enough to just check when you release the T cells into the blood. You also have to find ways of continuously checking afterwards that you're still not attacking your own cells. This was a very interesting way to do this experiment, because what they did is they took a peptide that came from the Y chromosome. Now, of course, you know the Y chromosome is only in males. So they looked for T cells that detected this peptide from a Y chromosome, compared how many they saw in males versus how many they saw in females. Females, this is a completely foreign peptide because, you know, it's not there in the genome, and therefore it wouldn't have been selected against. In males, it's a self-peptide, and so it should have been deleted. And the answer is, well, it's deleted, but it's deleted by about a factor of 10, maybe 5 to 10. It's not completely deleted. There still are cells that detect this peptide, even though it's, you know, should be completely deleted. So they call this, the paper was called Does Not Eliminate Specific Things, and this paper was called Not So Negative Selection, which was a nice way to put it. Now, this has a very important consequence for modern cancer therapy, because it says that you have higher levels of checking. And the higher level of checking got a Nobel Prize for this person, Jim Allison, a couple of years back, and made cancer immunotherapy the breakthrough of the year a couple of years back. And the idea is that there are other ways in which the T cell is prevented from attacking things that look like ourselves. I'm not going to describe this in detail, but there are other, you know, things that happen not at the molecular level, but at the cellular level, ways in which signals are exchanged between cells, and tell the cell that even though you look like you're detecting me, really I'm okay. So I have this cartoon, which I've always liked, which is sort of the idea that the cancer cell is playing the role of this, you know, would-be elk in the Yellowstone National Park, pretending not to be a bear. And Jim Allison found a way to disable this type of checking. Now, that's very dangerous, if you think about it, because you disabled something that is there to prevent autoimmune disease. But, you know, on the whole, if you do it the right way and you titrate the data, the drug concentrations well enough, you can find ways such that, you know, you more or less, you know, kill the cancer, and the disease, you know, the extra side effects from increasing your risk of some type of autoimmune disease are there. They do cause problems, but on the whole are better to deal with than having metastatic disease to kill you within 6 months. So this works for a number of cancers, and works, and this whole pathway, this whole idea was, as I said, due to Jim Allison and coworkers, and won the Nobel Prize in biology a few years ago for this method. And it's sort of built on the structure of saying you need to have these higher levels because you can't both detect tumor neoantigens and do a perfect job of discriminating against your own. Now, the last part I want to mention is, so why doesn't immunotherapy answer everything? Why doesn't now every single tumor be cured by the immune system, you know, given that we know how to make the immune system active and detect cancers? What's the problem? Why is it only that some cancers work with immunotherapy and some don't? So that's the last part of what I wanted to say, which is this picture over here, which I stole from a drug company, which is that sometimes, you know, there is physics that you have to worry about. You know, so far I've talked about networks and about signals and about information and about, you know, molecular interactions, but sometimes there's also physics. What do I mean by physics? Well, here is a series of pictures of tumors, and in these pictures, this gray region here is the tumor, and these brown dots are T cells. And these are actual patient tumors. So they're patient tumors taken out during surgery that are then stained such that you can detect the tumor and you can detect the T cells. And what you see is you see three different possibilities here. you see this possibility here, which they call the immune desert. The immune desert is, here's the tumor, and there are no T cells anywhere around. So this is a failure to detect. Right here, the immune system does not realize that this thing here is not supposed to be there. It's totally ignored the tumor. Okay, if that's the case, you're going to be in trouble, because your only choice then, and people are investigating this choice, your only choice then is to find ways of injecting new immune cells that will recognize that tumor. And people are designing new T cells. They're taking T cells and changing them and reinserting them in the body to try to accomplish getting problems like this, where your immune system just has failed to detect that this is a tumor. Okay. Now this is the case you want to see. This is the case where you have the tumor and the T cells are roaming freely throughout the tumor. And if they're roaming freely throughout the tumor, when they happen to hit a tumor cell and they happen to figure out that it's a tumor cell and they happen to be given this drug treatment based on Jim Allison's work that says, well, this thing might look like it's pretending to be not a tumor cell, but ignore that signal. It really is a tumor cell. Go kill it. In this case, you actually have a decent chance of immunotherapy working. This is sort of the interesting intermediate case where physics is a problem. Here you have a tumor, and somehow there's some barrier at the edge of the tumor, which is preventing the T cells from migrating into the tumor. So you see the T cells are congregating in the vicinity. They've detected that there's something wrong here, but if they don't make it into the tumor, they can't kill the tumor cells. Killing is a contact sport. You've got to actually attach to it. You've got to inject it with something to kill it. And here they just can't do it. This is called immune excluded. And you can see in this slide from this drug company, the different types of tumors. So to just translate, this is colorectal tumors, and you see that 12% is like this, 63% is like this. So you see that you won't expect immunotherapy to work very well for colorectal tumors. This is non-small cell lung cancer. This is much better. Here it's up to 31. You still have 44% here. This is triple negative breast cancer. This is, I don't even know what that is. And so you see that there's a large percentage here where somehow there's a physical barrier preventing the cells the tumor cells from being in contact with the immune cells. Now, what's going on? Well, we don't know. This is what some of us are working on. There's interesting papers that try to explain this, really thinking about physics in some way. So here's a paper that tries to explain what's going on. I don't know to what extent I believe this paper, but it's the sort of working hypothesis, at least several of us trying to figure out what's going on. This is the last slide I have, which is, here is what a cell looks like when it's moving around the tumor. Now, in those pictures, you see the tumor, you see the T cells, and then you see nothing outside. It's like the T cells are moving through outer space, with nothing there. They're moving through a vacuum. Well, the cells there's not a vacuum between our organs and tissues. There's space that's filled up with fibers that look like these fibers here. We have a space which is called extracellular matrix. It's filled with fibers of various proteins, and cells, in order to move through there, have to navigate their way through a very tortuous geometry. They have to push against this fiber network, and they have to make their way through the holes, and in order to enter, they have to make it through this jungle of fibers. Now, it turned out in this paper, published in Nature about 2 years ago, that the tumor somehow has arranged in ways that they don't understand and we don't understand, but nonetheless they show, has arranged such that this fiber network around the tumor is, first of all, very dense, and second of all, oriented to try to push the immune cells to move in a parallel direction to the surface of the tumor. So in other words, the T cell starts to move in, and they get redirected to move in the other direction. Instead of going this way, they go this way, and they can't go this way because of this very dense structure in between, and if the tumor cells can create this structure, they can prevent this thing from going in. Now, the good news in this paper for these people, if this turns out to be true, is that this is not always the structure outside the tumor. This is the structure sometimes outside the tumor. They claim they understand partially what the tumor needs to be doing to create this structure. There's some particular protein which is secreted that binds to this fiber, rearranges it as some type of phase transition, changes its structure, and causes this exclusion. If this is true, then of course one can find ways to counteract this. One might be able to reverse this exclusion if one understands better the motion of cells through this, the creation of these networks and their elasticity. And this is sort of showing that, there's still a role for physics to play in these problems. This is now no longer a signaling problem, no longer a detection problem. Now it's really just a cell motility and mechanics problem, which is not solved yet. So that's actually the forefront of sort of at least my thinking about the problem. Okay, so I'm done. So just a summary of what I said. So cancer cells are able to apply a number of adaptive strategies to avoid being destroyed by drugs, to avoid being destroyed by the human immune system. These arise by freeing the cells from regulatory constraints that arise whenever they, in evolution, created a multicellular organism. The immune system is very good at detecting things, but detecting cancer is much harder than detecting viruses and bacteria because, after all, in the words of this famous cartoon, we have met the enemy and he is us. Cancer cells look just like us, so you really need a very strong discriminatory system to actually tell the difference. And that the fact that it works at all is a testament to evolution of our immune system, but it's not perfect. It needs help from us. We showed how to help it at one level by giving these signal suppressing drugs that won a Nobel Prize. We have to show how to help it at the next level by actually allowing access to the tumors in cases where that isn't going to happen naturally. And then finally, the idea here is that, physical science has a role to play in many biological problems, not just cancer, but its role in cancer has been, let's say, slower in coming because of the complexities of the problem. I started working on biological systems on bacteria and on single-cell organisms, and the questions were much simpler. And here, everything is really complicated. The data is complicated. The system is complicated. The heterogeneity between different tumors, different patients, different parts of a tumor is, very difficult to deal with. But nonetheless, at the end of the day, combining experiments, measurements, clinical ideas with some type of computational physics, engineering-based strategy really, in my opinion, can offer insights to what's going on. And I view this as sort of a statement about that the way to make war on cancer is to be smart about it. And thank you very much for your attention. Yes.
Thanks for a great talk. Had a few questions.
Sure.
I won't try to take up
Sure.
all the time, but so the first one was in the cartoon that you showed in the first part of the talk of the mesenchymal
Yes.
cells and the transition. It seemed to be implied from the pictures that they are kind of structurally different?
Yeah, they are structurally different. I mean, when I talk about this to a more technical audience, I list, so these cells are very different from each other. They're as different from each other as our neurons are from our kidney cells.
Okay.
So neurons and kidney cells are a good example because they have the same genome, but yet they're structurally very different. They have different sizes, they have different shapes, they have different mechanics. So going from epithelial to mesenchymal is a transition which changes almost everything about the cells. It changes how they stick to each other, it changes their shape, it changes their symmetry. Epithelial cells are always, oriented sort of, they sit on the bottom of a membrane and they point upwards. It's called apical basal polarity. These cells lose that symmetry axis. They change their metabolism of how they use nutrients. So it's a complete change in the cell, which is why it's so hard to do and why it's amazing that some cancer cells can do that transition, but it is something that was engineered to be able to be possible in this developmental context.
So sort of a sequential question to that is that then you showed this work, I guess, with Mohit and so on, in which you had this intermediate thing which was much more efficient at metastasize.
Yes.
So then why doesn't it stop there? And if
well, I mean, it doesn't, the cell's not making a choice to stop there. In other words,
but isn't there some selection mechanism?
So, I mean, the answer seems to be that depending, so you might ask, why are they doing this in the first, what's telling them to do this? So it turns out that there are signals in the environment that convince them to undergo this transition. And for reasons that we don't fully understand, but have some ideas about, some cells are able to undergo this transition completely. But actually, the majority of the cells, in fact, do some version of doing this partially. And so that depends on sort of how changeable the cell's architecture is. And cells seem to come in a variety of changeability levels of their architecture. Some of them go partially through this, and some of them go completely through this. So it isn't like we're saying cells can't go completely through this. And even in most cases they go completely, they probably even go through a stage that looks like this and root. But it's the ones that stop in the middle that seem to be the ones we should worry about.
Which are more dangerous. So when you say that they are able to metastasize more, is it that somehow you measure that by their motility?
No, so it's not. So it's not really. I mean, so the point is, it's not really the motility, because motility, you can argue which is more efficient, moving individually or moving collectively, and it probably depends on exactly what geometry you're moving through. But the really hard part of metastasis, the part that usually is the one that is the most difficult, the rate-limiting step, is really starting to grow in a new environment. So it's the adaptability to go to a new environment and starting to grow there. And that you can test in the lab. You can take cells from these different, in vitro originally, you took these cells and put them in mimics in the lab, and so put them in 3D matrices that mimic some environment. And you can see that the ones in the middle are the ones that grow, they're able to start new tumors. And then that group was the first one to do that in a mouse experiment. And then there's later papers that did it in a pancreatic cancer mouse experiment. So it's really the ability to initiate growth and to go back into a sort of cell that's really adjusted to its environment and now growing as if it were back to a primary tumor. That really is the part that's hardest to accomplish, and that's the part. So it's not really the motility per se. I have this argument all the time with people who always work on motility, because I work on motility too, and I say cells can be motile in a number of ways. Maybe it's more efficient to transport by going into this purely mesenchymal route, but in the end of the day, that's not the hardest step. The hardest step is the regrowth, and that's the part that these cells are the best
much better at.
Yes.
Sorry, and then the very last question is, so in the last part of your talk where you talked about the immune action, if I understood correctly, it was mostly to do with the epithelial cells in the tumor as it is.
Yeah, in some sense. Yeah,
I was just wondering, are there strategies which try to attack the intermediate, these mesenchymal or the intermediate stages, at least to prevent it from metastasizing?
So I think it's an open question about how the ability of the immune system to attack cells depends on whether they're epithelial or mesenchymal. In other words, really what I was, especially this invasion picture, was really focused on the primary tumor, right? In other words, the immune system has no trouble if you have isolated cells in the blood system, the immune system can attack them. So there are cases where the immune system is not successful in attacking the primary, in mice, of course, what you can do, is not successful at eliminating the primary tumor, but does prevent metastasis. So there are cases like that, because they have access to the cells as they pass through the blood system. That's where the T cells are anyway. So I didn't talk about that. That's a separate question, but normally when people think about when the immunotherapy language is always done in terms of eliminating all the residual cells around the primary tumor, and for that case, it's sort of like the epithelial cells. But some of those questions are not known.
I mean, because I was wondering whether medical intervention could be that you sort of just surround it and prevent that region from, in a sense, growing, and then use something else like chemotherapy or the radiation or something to destroy the primary.
So I mean, that's what people do try that. But I think the answer is we don't quite know. I mean, there's some evidence, although, you know, I would say the jury's not completely settled, although maybe Mohit disagrees with me, that the cells that are able, that these partial EMT cells that are able to adapt to get, you know, to sort of win out against, you know, drug therapy in addition to being able to metastasize, are also better able to evade immune cells. There's some evidence of that, but it's not complete. So right now, the focus has been just on attacking the primary tumor. That's what everybody's been thinking about. It would be fine, as you say, if we could figure out how to get the immune system to just prevent metastasis, and then we'd have other techniques to do that. That doesn't seem to be what typically people are getting to work yet. People are, anyway, as far as, you know, so I think it's just too early to understand that. Anyway, that's not a very satisfying answer, but I just have nothing more satisfying to say. Yeah.
Cells that have this dense matrix.
Yes.
Hello. Yeah.
Oh, yeah.
So when they have dense matrix, how is it affecting the metastasis in those cancers? Like, is there data available for that? Have you checked?
So it's sort of funny because, you know, there was originally papers in the breast cancer literature which argued that having the fibers arranged sort of circumferentially around the tumor was actually good because it prevented the tumor cells from leaving. So the idea used to be in the breast cancer world that there was even this thing that was an attempt to make a clinical correlate of that called the tumor-associated collagen signature, which basically said your chance of getting metastasis from a breast cancer would be lower if your network was arranged circumferentially around, you know, the tumor, because if a cell tries to leave, it would be sort of trapped in the network to sort of not be able to move further in the radial direction. So now the argument is exactly reversed. Now the argument is, well, that's true, but it's preventing the immune cells from entering. So it's probably doing both. So in the experiment that is in this Nature paper, they didn't focus on metastasis. They focused on the ability of the immune system to attack the primary tumor. It's sort of like the question we had before, and it prevented the immune system from attacking the primary tumor. They didn't report one way or the other because they just weren't focused on that. If you know what was happening in terms of whether those tumor cells were metastasizing. So it looks like this organizational structure is both helping and hurting at the same time, at least according to what was published in the breast cancer literature earlier.
Thank you.
No?
Yeah, hi. So I was just wondering if the metastatic or the mesenchymal cells, they migrate via the bloodstream,
Yes.
then why is it not that, or why is it such that certain cancers metastasize to certain locations?
Yeah.
Like, why is it not random?
Okay. Yeah,
Is there a reason?
So the good thing about giving a talk about cancer is that you're always one step away from the unknown answer. So first of all, the answer is not distance through the blood system. It's been established in very great detail that it isn't a question of where you get to first. So it really is what used to be called, or maybe still called in the cancer literature, matching the seed and the soil. That was their language. That somehow the certain types of cancer, let's say breast cancer, somehow have less adaptation to do to metastasize to the bone than they have to metastasize to, I don't know, what's a good example where they don't metastasize to, the pancreas, I don't know. So exactly what that signature is and how to, and predicting that, I don't know of anybody who's made a serious prediction of that. Is anybody? You don't know either. Good, okay, it's not just me. And even within breast cancer, I think we, in one of the talks we heard today, breast cancer of certain subtypes, the ones that are usually called hormone-positive breast cancers, the ones we can treat, they typically will metastasize to the bone, maybe a little bit to other places, but at least the bone is a classic place. And like triple-negative breast cancer, which doesn't have this, somehow metastasizes to the brain and to the lungs. So it's still breast cancer. They started out from similar cells in the breast, but there's different enough that somehow they have this propensity to be much more effective in one place or the other. It is partially evident that this is one of the reasons we think that the growth part is the most critical part, because without the growth part being the most critical part, you would say this is crazy, right? If making it through the blood system is the hardest part, then wherever you would go, you would be able to metastasize. But it's not true. Metastasis is very organ specific, and I don't think we understand that. I think one can hope that these concepts of your adaptation to your new environment, coupled with a better understanding of what is it in the environment that the cells are very sensitive to, maybe we'll be able to answer that question in the near future. But right now, there's no answer. It just is true.
Really lovely talk. My question was, do we understand why these intermediate state cells are the ones that are better capable of adapting to new environment? Is it like?
Do we understand why?
Yeah.
So the original explanation of why was that there's a picture. Maybe I'll use the blackboard since there's
Like
so
a
many blackboards
metastable
here.
state,
There's
so it falls down.
Yes, exactly. Yes. So the original picture was that it was sort of reminiscent of a developmental biology pathway. So in a developmental biology pathway, you have a progenitor cell, and then you have, this will then differentiate into cell of type A and a cell of type B, which will be fully differentiated cells. So for example, in the hemopoietic, the white blood system, you have many cases like this type of bifurcation. You start out with something which is a multipotent cell, and it then decides to go, either a neutrophil or whatever, macrophage, whatever it is. So the idea was that at a conceptual level was that the same picture was being reproduced in this EMT language, that you had this E cell here, which was very differentiated. You had this M cell here, which was a very specialized differentiated cell. And to go from one to the other, you went up here to a cell which was less differentiated, which was E/M. Now, the evidence for this picture, besides the fact that it sort of fits conceptually, was that if you look at what was predicted for the gene signature of this state, in particular, you look at various factors which are associated with stemless plasticity, things like OCT4 or NANOG or whatever, it turns out that the circuits are such that these cells have higher levels of those stem-like cells. So if you try to make a stem-like signature, then you discover that these cells are the ones that contain all of those stem-like factors. So just like these are sort of, less differentiated, they are more stem-like, they're closer to, original embryonic stem cells, and they're just sort of moving their way down. This cell had the gene signature predicted to be the same and then measured to be the same. So it was because of the predicted gene signature being closer to stem-like cells that one thought of this. And then there was this conceptual mapping that sort of explained things like this. So that was the logic. And that seems to be what happened. That seems to still be true. It was proposed, I don't know, what year did we propose that? 8 years ago?
Yeah, 10 years ago.
10 years ago? Well, time goes by, it's gonna be fun. Yeah.
Cells would be the ones that would be able to survive.
No, the E cells are more specialized for sticking to each other, sticking to the basement membrane, and when they need to, growing faster. I mean, they say sometimes are turned off, but if you give them growth signals, they'll be able to grow faster also. So these cells are not the ones that are most adaptive. They're the ones that are most effective at growing when you already have a sort of established colony. So the idea of starting something new is the hard part. These cells, like to stick to each other. They like to stick to a basement membrane of a certain kind. If you don't have those things, these cells are lost. Once these cells start growing, then they start making those things, they start making other cells around them, and eventually they might be able to convert back to E cells to grow more efficiently. But that doesn't matter. These are the ones that are able to start the new growth in this model and in the experiments that we reported.
So we'll take one last question, and then there's tea outside, so maybe if people have other questions, you can ask them that.
Okay, this is like more comment than question. Like somebody asked organ-specific metastasis, right?
Yes.
So I think cancer cells, what also we observe here in ER positive and triple negative breast cancers, they have special receptors, and the organ like liver is actually secreting certain ligands for that. So it creates a kind of a navigation system with that receptor that binds, and then it gives a preference for proliferation at that site. Suppose HGF receptor is present in many breast cancer once they start to be EMT, like EMT like. So HGF is actually secreted from the liver, and it gives a very nice homing to that organ, so that with HGF receptor on tumor cells, which is EMT, and HGF being secreted by the liver cells, so it provides a nice home for that, and the tumor can then settle down into the liver and can start proliferating. And it happens in several type of organs. What a tumor cell has as a receptor, and what is secreted by any other organ like bone, it secretes SDF, some other ligand. So ER positive cells has more of SDF receptor, so they start to move towards that Q, and then they settle down at the bone. TNBC on the counterpart, they have more HGF which settle down in the liver. So a lot of research has been going into this area, that how a tumor cell can home into different kind of organs. And it looks to us that a lot of cytokines, chemokines, growth factors are involved into that navigation, as well as homing and seeding to that metastatic niche. So it happens like that. And even we have seen in our systems, when the cell become EMT, certain special receptor which was never present before in the epithelial cells, now they start to express. So it actually explains what is happening. Thank you.
So I think with that, let's thank Herbie once again for an excellent talk.
Oh, okay.